Hi, hello, my dear students. This is your Prasanna Harikrishna sir, Assistant Professor of Botany, SRR Government Arts and Science College, Karimnagar. So today we are going to discuss about photosystems of photosynthetic process. In the last session, we have seen red drop phenomenon or red drop process. Clear? So today, we'll discuss the entire picture of photosystems. Clear? So I will draw the one particular diagram. You can easily recap the earlier concepts. For example, this is a plant, right? So every plant has root system, stem and the leaves. Clear? So these are the leaves. This is sunlight which penetrates on the leaf. Then it is useful for performing photosynthetic mechanism. Clear? So when you see the composition of the leaf, for example, this is the composition of leaf, right? So every part or every living organism or every part of the living organism is made up of cells. For example, these are all the cells. Group of cells is generally called as tissue. Okay. For example, I isolate a group of cells. This can be called as what? Tissue. Okay. This is what? Tissue. Then I separate one single cell from the tissue. This is one single cell. Clear? So in the cell, for example, if you go inside, right? If you go inside, there is a liquid substance that can be called as what? Protoplasm. Or you can say protoplasm is divided into two basic components. One is cytoplasm and the nucleoplasm. Clear? So for your understanding, this is entire the liquid substance, cytoplasm. Okay, there is a nucleus inside the cell. Apart from nucleus, this cytoplasm is having different kinds of smaller substances or smaller structure. Every smaller structure which is there in the cytoplasm can be called as what? Cell organelle. Cell organelle. Cell organelle is the smaller or specialized structure which are present inside the cytoplasm of the cell. Clear? One of the cell organelles is chloroplast. For example, these are all the chloroplasts which are located in the cytoplasm. You might have heard mitochondria, endoplasmic reticulum, lysosomes, ribosomes, etc., etc. So they are all called as cell organelles. Okay. So in our context, we are going to see in detail structure of chloroplast. Clear? Then if you isolate one particular chloroplast from cell, this is what? Chloroplast. I have already told you the structure of chloroplast. Then inside the chloroplast there is thylakoid. Group of thylakoids can be called as grana. Right? Now I isolate one particular thylakoid. This is what? Thylakoid structure. Okay. So in the middle of the thylakoid, this is middle or inner portion of thylakoids. This entire inner portion of thylakoids is acting as the reservoir for water. It is having abundant water molecules. Clear? This inner side of this particular thylakoid can be technically called as lumen. What is that? Lumen. Okay. So this is outer layer, this is the inner layer. In between these two layers, there are different kinds of systems. There are different kinds of systems. For example, if you see the diagrammatic representation, these are the systems. Okay. They receive sunlight. These systems receive sunlight. Okay. So these systems can be differentiated into, these systems are called as what? Photosystems. Whenever you hear the term photo indicates what? Light. Light means sunlight. These systems receive sunlight, hence these systems are technically called as photosystems. Okay, every photosystem, remember, every photosystem, for example, this is a system. This system is composed with chlorophylls. What is that? Chlorophylls. Chlorophylls are the green colored pigments, and these systems are composed with xanthophylls. Xanthophylls or yellow colored pigments. Then these systems are also composed with carotenes. Carotenes or orange and red colored pigments. Okay. So every system receives light. Hence they are called as what? Photosystems. And every system is made up of, or you can say is composed with different kinds of color giving substances or pigments. These pigments include chlorophylls, that is green color pigments, then xanthophylls, yellow color pigments, then followed by carotenes, orange and red colored pigments. Hence every system is composed with pigments or color giving substances. Hence the alternative name to photosystems is pigment systems. What is that? Pigment systems. So this is an alternative name to photosystems. Then now we are going to see the clear-cut differentiation of these two photosystems. Generally, according to Robert Emerson, in earlier class we have discussed about red drop mechanism. So red drop mechanism was given by whom? Robert Emerson. So Robert Emerson identified two kinds of systems, or you can say photosystems, in the thylakoid membranes. One is PS1 and the second one is PS2. So PS stands for what? Photosystem, or you can say pigment system 1, and pigment system or photosystem 2. Clear? So now we are going to discuss, or we are going to see the complete differences between these two systems. Now I want to tabulate the information for clear understanding. This is PS1. This is PS2. PS1 stands for photosystem 1, PS2 stands for photosystem 2. Right. Every photosystem is there in thylakoid layers. Clear? Now, this PS1 has the reaction center. PS1 reaction center can be P700. Okay. PS1 reaction center is what? P700. Then PS2 reaction center will be 680. That is P680. Okay. So P700, 700 indicates what? So P700 indicates a chlorophyll molecule that is very active when compared to another chlorophyll molecules. For example, this is what? Photosystem. Okay. Now I isolate this photosystem. This is a photosystem. It is composed with different kinds of pigments. Okay. These are all the pigments. Apart from all the chlorophylls, carotenes and xanthophylls, one of the chlorophyll molecule is very active when compared to another chlorophyll molecules. That particular, for example, for your understanding, I am quoting this particular chlorophyll molecule. This chlorophyll molecule is very active when compared to other chlorophyll molecules. So this can be called as reaction center. What is that? What is meant by reaction center? Reaction center is a chlorophyll molecule which is very comfortable, which is very active when compared to another chlorophyll molecules. Clear? So in this context, PS1 reaction center is P700. P700 nothing but what? This is a reaction center. It receives, this particular reaction center receives 700 nanometers of sunlight. Okay. So then come to PS2. In PS2, what is the reaction center? P680. P680 nothing but what? The chlorophyll A molecules receive 680 nanometers of sunlight. Simple. Then come to next level. Exciting state. Exciting state is considered as chlorophyll star. That is chlorophyll 700. Exciting state is denoted in the form of chlorophyll CHL star 700. What does it mean? For example, this is a chlorophyll molecule. Clear? This is a chlorophyll molecule. I'll draw here. This is a molecule. Okay, so every molecule has, outer orbits. According to chemistry, according to atomic structure, every molecule has, outer orbits. Okay, so when the molecule receives energy, so what kind of energy here? Sunlight, sun energy, that is, heat energy is received by this particular chlorophyll A molecule. Whenever it receives energy, the outer orbital electrons are there in the outer orbital, right? So these electrons will be excited in condition. Okay, this molecule will be excited in condition, and finally the electrons are lost after receiving energy. Okay, so electrons are lost when the molecule becomes excited. When the molecule becomes excited, when it receives energy. What kind of energy in this particular context? That is, heat or light energy. Okay, when you give light energy to chlorophyll A molecule, the outer orbital electrons will be lost. Clear? That condition can be called as what? Exciting condition. It denotes in the form of CHL star. Chlorophyll star. Star indicates what? Exciting condition. What is a molecule here? Chlorophyll A molecule, that is 700. Chlorophyll 700. What about this one? Simple. Exciting condition of PS2 is chlorophyll star 680. Okay, simple. Then this PS1 has carotenes as accessory pigments. Remember, I have already told you in my previous classes. What is accessory pigments? Accessory nothing but additional or protective or supportive pigments. For example, these are all the chlorophyll molecules. Okay, these chlorophyll molecules are surrounded by different pigments. These are all the different pigments which surround chlorophyll molecules. Clear? So these surrounded or accessory or supportive or protective pigments are carotenoids. So carotenoids are categorized into two basic pigments. One is xanthophylls and the second one is carotenes. Okay, so carotenoids include xanthophylls or carotenes. Clear? So but PS1 only carotenes are present. Okay, so carotenes are present in the PS1. Then come to PS2, xanthophylls are present. Okay, so xanthophylls are present in PS2. Simple. Both carotenes and xanthophylls are included in carotenoids. Clear? So carotenes are red and orange colored pigments, whereas xanthophylls are yellow and brown colored pigments. For example, if you see tomato, the ripened tomato is in red color. The ripened tomato is in red color due to having carotenes. There are different kinds of types in carotenes. For example, with respect to tomato, there is a lycopene. So lycopene is a pigment, basically a carotene, which is present in the tomato. So due to lycopene pigment, tomato or ripened tomato color is red. Then you can observe another example, that is ripened chili. So ripened chili is in red color due to carotenes. Whenever you or wherever you see red or orange color in the plants, the responsible pigment will be carotenes. For example, orange, carrot. Carrot is in orange color due to beta carotene. So beta carotene is a orange colored pigment. Nothing but what? Basically beta carotene is carotene. Okay, then when you see xanthophylls, wherever you see the yellow color or brown color in the plants, the responsible pigments will be xanthophylls. Okay, then this chlorophyll A and B. So chlorophylls are categorized into 9 types according to Arnon, right? According to Arnon, entire chlorophylls are categorized into 9 types. Okay, out of 9, the dominant chlorophylls are chlorophyll A and chlorophyll B. Okay, along with chlorophyll A and B, chlorophyll C is there, D is there, E is there. And bacterial chlorophyll A, bacterial chlorophyll B, chlorobium chlorophyll 650, chlorobium chlorophyll 660. So these are the 9 types of chlorophylls, but in plants these two are more abundant, chlorophyll A and chlorophyll B. The ratio between, for example, these are all the chlorophylls now. Here chlorophyll A is there, B is there, A, some of the chlorophylls are A chlorophyll, some of the chlorophylls are B chlorophylls. The ratio between chlorophyll A and chlorophyll B with respect to photosystem 1 will be 4:1. Remember, what is the ratio between chlorophyll A and B? 4:1. Means if 4 chlorophylls are A type, 1 will be B type. Okay. So for example, if 5 chlorophylls are there in the photosystem 1, apart from 5 or out of 5, 4 will be chlorophyll A, 1 will be chlorophyll B. Then come to photosystem 2. The ratio between chlorophyll A and B will be 1:1 only. Okay, means what? If 1 chlorophyll A, there will be 1 chlorophyll B. But the ratio differs from PS1 to PS2. Remember, this kind of information can be asked in the competitive examinations like PG entrance exams. Remember. Okay. Then the thylakoids are isolated thylakoids. Okay. Isolated thylakoids are found in this particular system. Means here group or grouped thylakoids are there in photosystem 2. Photosystem 2 is found in grouped thylakoids. Photosystem 1 is found in isolated thylakoid. Isolated nothing but what? Separated. Single. For example, this is the chloroplast. This can be called as a group of thylakoids. For example, if you observe one single thylakoids in the entire picture of chloroplast, this can be called as what? Isolated, or you can say isolated or single thylakoid. So this entire portion can be called as what? Grouped thylakoids. Okay. Then the redox potential. Remember, the redox potential of PS1 will be plus 45. Clear? Redox potential of PS1 will be plus 45 and the redox potential of photosystem 2 will be plus 82. Plus 45 plus 82. Then come to RP, that is redox potential. What is meant by redox potential? Clear? So for example, this is what? Molecule. Okay. Every molecule or every component loses the electron and receives the electron. Generally, the tendency of every molecule is having two kinds of properties. For example, this is a molecule or this is a substance. It may receive electrons. Okay. It may lose the electrons. Okay. Some of the substances receives the electron, some of the substances loses the electron. Of course, one single substance receives the electron, that substance will lose the electrons as well. Clear? So this property, receiving and losing the electrons, this property can be called as what? Reductive potential. Okay, or reducing potential you can say. Okay, so loss of electrons and receiving of electrons is called as reducing or redox potential. Okay, so this redox potential is plus 45 with respect to photosystem 1. Redox potential of photosystem 2 is plus 82. Then come to the number or numerical or statistical information. So if you observe the statistical or numerical information, I added plus before 45. Remember, RP always indicates in positive or negative manner. If there is a plus symbol before, any numerical information or any numerical number, this indicates what? Plus always adding. Plus means always adding. Adding nothing but electrons are received. Whenever the substance or molecule receives electron, that can be, called as plus or positive or addition, you can say. Okay, I am using plus 45. Plus 45 nothing but what? This photosystem 1, the entire structures of photosystem 1 receive the electrons, and photosystem 2 also receive the electrons. The redox potential is more than this is just plus 45, this is plus 82. Number means what? The redox potential of photosystem 2 is more than the redox potential of photosystem 1. Okay, so if the number, for example, if you see minus 30, whenever you observe the number minus 30, what it indicates? There is a minus before the number or numerical data. So minus indicates what? Losing or loses the electron. Whenever electron is added, that can be called as plus or positive or addition. Whenever the substance loses the electron, that denotes in the form of minus. Okay, so this is for your understanding. Just remember, RP of PS1 is plus 45, RP of PS2 is plus 82. Then the final concept of this particular differentiation between photosystem 1 and 2 will be the first electron receiver. The first electron receiver of PS1 is pheophytin. So pheophytin will be the first electron receiver of pheophytin. I can tell you in detail, that is iron sulfur protein. What is that? So iron sulfur protein will be the first electron receiver of PS1. Then on the other hand, here pheophytin is the first electron receiver with respect to photosystem 2. Clear? So this is all about differences between PS1 and PS2. Clear? So in next session we will discuss photophosphorylation. So photophosphorylation is categorized into cyclic and non-cyclic photophosphorylation. Right? So thank you one and all.